this video we are going to learn about apoptosis, that is the programmed cell death and its process, different modes, what is importance, what happens when some difficulties occur in the process of apoptosis. All these things we will be learning with the help of this video. So, apoptosis or programmed cell death is a normal process that is unique to animal cells. Apoptosis occurs through an orchestrated sequence of events that lead to the death of a cell. Death by apoptosis is a neat, orderly process characterized by the overall shrinkage in volume of the cell and its nucleus, the loss of adhesion to neighboring cells, the formation of blobs at the cell surface, the dissection of the chromatin into small fragments, and the rapid engulfment of the corpse by phagocytosis. Apoptosis is often contrasted with a different type of cell death called necrosis, which generally follows some type of physical trauma or biochemical insult. Like apoptosis, necrosis is generally considered to be a regulated and programmed process, although much less orderly in nature. Necrosis is characterized by the swelling of both the cell and its internal membranous organs, submembrane breakdown, leakage of cell contents into the medium, and the resulting induction of inflammation. Because it is a safe and orderly process, apoptosis might be compared to the controlled implosion of building using carefully placed explosives as compared to simply blowing up the structure without concern for what happens to the flying debris. Why do our bodies have unwanted cells, and where do we find cells that become targeted for elimination? The short answer is almost everywhere you look. During embryonic development, the earliest form of human hand resembles a paddle without any space between the tissues. That would become the fingers. The fingers are essentially carved out of the paddle via the elimination of the excess cells by apoptosis. The three stages of this process are very important. T lymphocytes are cells of immune system that recognize and kill abnormal or pathogen-infected target cells. These target cells are recognized by specific receptors that are present on the surface of T lymphocytes. During embryonic development, T lymphocytes are produced that possess receptors capable of binding tightly to proteins present on the surface of normal cells within the body. T lymphocytes that have this dangerous capacity are eliminated by apoptosis. Apoptosis does not stop at the end of embryonic development. It has been estimated that 10 raised to 10 to 10 raised to 11 cells in the adult body lie every day by apoptosis. For example, apoptosis is involved in the elimination of cells that have sustained irreparable genetic damage. This is important because damage of the genetic blueprint can result in unregulated cell division and the development of cancer. Apoptosis is also responsible for the death of the cells that are no longer required, such as activated T cells that have responded to an infectious agent that has been eliminated. Finally, apoptosis appears to be involved in neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and Huntington's chorea disease. Elimination of essential neurons during disease progression give rise to loss of memory or decrease in motor coordination. These examples show that apoptosis is important in reshaping tissues and organs during embryonic development and in maintaining homeostasis within the bodies of adult animals. Serious diseases can result from both the failure to carry out apoptosis and the elimination of cells is appropriate, and form the overreactive induction of apoptosis when the elimination of the cell is not appropriate. For example, type 1 diabetes. The term apoptosis was coined in 1972 by John Kerr in the University of Scotland in a landmark paper that described for the first time the coordinated events that occurred during the programmed death of a wide range of cells. Insight into the molecular basis of apoptosis was first revealed in studies on the nematode worm C. elegans, whose cells can be followed with absolute precision during embryonic development. Of the many cells produced during the development of the sperm, 131 cells are normally destined to die by apoptosis. In 1986, Robert Horwitz and his colleagues at the Massachusetts Institute of Technology discovered that worms carrying a mutation in the CED3 gene proceed through development without losing any of their cells to apoptosis. This finding suggested that the product of CED3 gene, CED3 gene, played a crucial role in the process of apoptosis in this organism. Once the gene has been identified in one organism, such as a nematode, researchers can search for homologous genes in other organisms, such as humans and other mammals. The identification of CED3 gene in nematode led to the discovery of a homologous family of proteins in mammals, which are now called caspases. Caspases are a distinctive group of cysteine proteases with a key cysteine residue in their catalytic site that are activated at an early stage of apoptosis and are responsible for triggering most, if not all, of the changes observed during cell death. Caspases accomplish this feat by cleaving a select group of essential proteins. Among targets of caspases are the following: more than a dozen protein kinases, including focal adhesion kinases FAK, PKB, PKC, and RAF1. Inactivation of FAK, for example, is presumed to disrupt cell adhesion, leading to detachment of appropriate cell from its neighbors. More than a dozen protein kinases, including focal adhesion kinases FAKs, PKBs, PKCs, and RAF1. Inactivation of FAK, for example, is presumed to disrupt cell adhesion, leading to detachment of the apoptotic cell from its neighbors. Inactivation of certain other kinases, such as PKB, serve to disrupt pro-survival signaling pathways. Caspases also disrupt the generation of survival signals by inactivating the NF-kB pathway. Lamins, which makes up the inner lining of the nuclear envelope. Cleavage of lamins lead to the disassembly of the nuclear lamina and shrinkage of the nucleus. Proteins of the cytoskeleton, such as those of intermediate filaments, actin, tubulin, and gelasolin, cleavage and consequent inactivation of these proteins lead to changes in cell type. So caspases accomplish this feat by cleaving the select group of essential proteins. Many of the targets of caspases are more than a dozen protein kinases, including focal adhesion kinase FAK, PKB, PKC, and RAF1. Inactivation of FAK, for example, is presumed to disrupt cell adhesion, leading to the detachment of the apoptotic cell from its neighbors. Inactivation of certain other kinases, such as PKB, serves to disrupt pro-survival signaling pathways. Caspases also disrupt the generation of survival signals by inactivating the NF-kB pathway. Lamins, which make up the inner lining of the nuclear envelope. Cleavage of lamins lead to the disassembly of the nuclear lamina and shrinkage of the nucleus. Proteins of the cytoskeleton, such as those of the intermediate filaments, actin, tubulin, and gelasolin, cleavages and consequent inactivation of these proteins lead to changes in cell shape. Now, an endonuclease called caspase-activated GNase (CAD), which is activated following caspase cleavage in an inhibitory protein. Once activated, CAD translocates from the cytoplasm to the nucleus, where it attacks DNA, turning into an active form which turns the DNA to fragments. Recent studies have focused on the events that led to the activation of a cell suicide program. Apoptosis can be triggered by both internal stimuli, such as abnormalities in the DNA, the external stimuli, such as certain cytokines, proteins secreted by cells of the immune system. For example, epithelial cells of the prostate become apoptotic when deprived of the male sex hormone testosterone. This is the reason why prostate cancer has spread to other tissues, is often treated with drugs that interference with testosterone production. Studies indicate that external stimuli activate apoptosis by a signaling pathway called the extrinsic pathway that is distinct from the and that one utilized by internal stimuli, which is called the intrinsic pathway. Here we will be discussing the extrinsic and intrinsic pathway separately. However, it should be noted that there are crosstalk between these pathways and that extracellular apoptotic signals can cause activation of the intrinsic pathway. The extrinsic pathway of apoptosis. The steps in the extrinsic pathway are given in the case depicted in the figure. The stimulus for apoptosis is carried by an extracellular messenger protein called tumor necrosis factor, TNF, for short representation, TNF, tumor necrosis factor. It is named for its ability to kill tumor cells. TNF is a trimeric protein produced by certain cells of immune system in response to adverse conditions such as exposure to ionizing radiation, elevated temperature, viral infection, or toxic chemical agents such as those used in cancer chemotherapy. Like other types of host messengers discussed in this chapter, TNF evoke its response by binding to a transmembrane receptor, TNFR1. The trimeric TNFR1 protein is a member of a family of related death receptors that turns on the appropriate apoptotic process. The cytoplasmic domain of each TNF receptor subunit contains a segment of about 70 amino acids called a death domain, each green segment in the figure, and that mediates protein-protein interactions. Binding to TNF to the trimeric receptor produces change in conformation of the receptor's death domains, which leads to the recruitment of a number of proteins. The last protein to join the complex that assemble at the inner surface of the plasma membrane are two procaspase 8 molecules. So TNF will be there, TNFR1 will be there, death domains are there, plasma membrane, then FAD along with that TRADD, procaspase 8, initiator caspase 8, and executioner procaspase along with the executioner caspase, and then apoptosis is happening. So when TNF binds to a TNF receptor, TNFR1, the activated receptor binds to two different cytoplasmic adapter proteins like TRADD and FADD and procaspase 8 to form a multi-protein complex at the inner surface of plasma membrane. The cytoplasmic domains of the TNF receptor, FADD and TRADD, interact with one another by homologous regions called death domains that are present in each protein, indicated as green boxes, and procaspase 8 and FADD interact by means of homologous regions called death effector domains. And once assembled in this complex, the two procaspase molecules cleave one another to generate an active caspase 8 molecule containing 4 polypeptide segments. Caspase 8 is an initiator complex that activates downstream executioner caspase that carry out the death sentence. It can be noted that the interaction between TNF and TNFR1 also activates other signaling pathways, one of which leads to cell survival rather than self-destruction. So these proteins are called procaspases because each is a precursor of caspase. It contains an extra portion that must be removed by proteolytic process to activate the enzyme. The synthesis of caspase as proenzyme protects the cell from accidental proteolytic damage. Unlike most proenzymes, procaspases exhibit a low level of proteolytic activity. According to one model, there are two main procaspases that held in close association with one another. As they are very closely associated, they are capable of cleaving one another's polypeptide chain and converting the other molecule to the fully active caspase. The final mature enzyme contains 4 polypeptide chains derived from 2 procaspase precursors, and activation of caspase 8 is similar in principle to the activation of effectors by a hormone or growth factor. In all of the signaling pathways, the binding of an extracellular ligand causes a change in conformation of a receptor that leads to the binding and activation of proteins situated downstream in the pathway. Caspase 8, described as an initiator caspase because it initiates apoptosis by cleaving and activating downstream or executioner caspase that carry out the controlled self-destruction of the cell as mentioned earlier.